Hi everyone, so today we're going to go over the process of transcription, and yeah, so essentially first things first, we want to understand that DNA is a double-stranded molecule, and it's coiled up in this helix type shape, as we can see right here. It's this kind of helix shape. Now when we look at the double strands, when we look at the two strands separated from each other, we know that one of these strands is called the template strand and one of these strands is called the coding strand. Well, the template strand is essentially what RNA polymerase binds to create a complementary sequence of. So RNA polymerase binds here, creates a complementary sequence to the template strand, creates complementary sequence. Yep. Now the coding strand, on the other hand, contains the specific genetic instructions or the specific DNA base sequence to create a protein. So let's just say that to create protein X, right, we want the sequence ACGGAC. Now this specific sequence is going to be embedded into our coding strand. This is the DNA bases on that is inside our coding strand, right? So what actually is the process of transcription? Let's just start now. So first things first, RNA polymerase binds to our template strand, right? Because remember, the template strand is where RNA polymerase binds to create a complementary sequence. RNA polymerase will only ever bind to the template strand. You will never find it binding to the coding strand, right? Unless, yeah, you will never find it, yeah. Well, RNA polymerase binds to a template strand, so let's just draw RNA polymerase binding to that. So what it essentially does, right, is that RNA polymerase binds to the primary sequence of our template strand to initiate transcription. Now, we know that this is kind of coiled together, right, in this, like, tight thing. So essentially what RNA polymerase will do is kind of unwind a section of it to, kind of, be able to do its process, because it can't do its process if the DNA is like the double strands are really tightly coiled together like in a helix, right? So RNA polymerase will like kind of let's just draw like it kind of unraveling. Yeah, so it will basically unravel a small section. It won't unravel the whole thing at once. It will unravel a small section, and yeah, that's how transcription starts. So in this small section, right, let's just say that this is a small section. We have RNA polymerase. This is the coding strand. RNA polymerase is this is the template strand. RNA polymerase is bounded to this. We're just drawing this section unraveled already. So let's just say that the coding strand here, and let's just like that. So the coding strand contains the sequence ACGGAC. So ignoring RNA polymerase for a second, the template strand will have the complementary sequence to the coding strand, because in DNA both strands have complementary sequences to each other, complementary base sequences to each other, right? So if the coding sequence has this strand, the template strand will have the opposite, which is A pairs with T, C pairs with G, G pairs with C, and so on and so forth. Yeah, so this will be the sequence on the template strand, and together these strands make up DNA. So what RNA polymerase will do, right, is now it will create, as we set up here, it creates a complementary sequence to the template strand. Essentially what it will do is that it will create a complementary sequence to this, right? So another thing, first things first, since our first base is a T, essentially there is one major difference between RNA and DNA in terms of bases, is that in DNA you have A, T, C, G. However, in RNA you will have A, U, C, G. So essentially T is replaced by uracil, thymine is replaced by uracil, yeah? So yeah, so we'll see, we'll get, this specific sequence doesn't involve anything like this, so we will see an example later on. But yeah, so we know that RNA polymerase will create the opposite to this, which is T pairs with A, G pairs with C, C pairs with G, G, A, C. So we can kind of already see that our RNA, pre-mRNA, sorry, matches exactly with our coding strand, and that's how you know that you've done it right, when your pre-mRNA strand matches exactly with the coding strand. Now there is a there is a situation where it's a little bit different. Pre-mRNA, strand is a little bit different to our coding strand, and that is what I'm going to be showing you. So for example, say to create protein Y now. Let's just say we want to create protein Y. The specific sequence is, yeah, so specific sequence is TAG GAC, right? Let's just say GAC. Now, if this is our coding strand, this specific sequence is obviously found in our coding strand, right? If this is our coding strand, our template strand will be the opposite to this, which is A, T, C, C, T, G. Yeah? Now what RNA polymerase will do, again, binds it, sorry, no, that's not the thing. It binds to the template strand, it creates a complementary to this specific sequence. So, what is it going to be? What is the RNA strand? Well, as we know, T is replaced by U, right? So, in this sense, we have to write down what A pairs with. Well, in DNA, we know A pairs with T, right? But however, in RNA, there is no T available, so A will actually pair with U, and then T pairs with A, C pairs with G, G, A, C. So in this sense, we can kind of see that our coding strand and our RNA template is a little bit different. It's just that the fact that wherever T is, U replaces that. So in this sense, it's a bit different, but yeah, that's all you need to know really about transcription. Transcription stops when a stop codon is reached and, you know, the pre-mRNA sequence is released from the thing, and that's basically all you need to know. It's a really, really easy and straightforward process. But yeah, if you have any questions, feel free to ask, and please do visit our website for more help. Bye bye.